Hello, my name is Falk Schlaudraff. I work as a product manager at Leica Microsystems. Today, it's my pleasure to introduce to you Thierry Nordmann and interview him on his recent research. Thierry Nordmann is a clinician and scientist in Munich. As a clinician, he works at the University Hospital of the Ludwig Maximilian University, and as a scientist, he is in the Matthias Mann group at the Institute for Biochemistry, so the Max Planck Institute for Biochemistry. Thierry and his team applied a technique called Deep Visual Proteomics, in short DVP, which combines high-resolution imaging, AI-based segmentation, laser microdissection, which comes from Leica, mass spectrometry, high-sensitive mass spectrometry, to analyze diseases on a single-cell level. He applied DVP to a lethal disease which is called TEN, toxic epidermal necrolysis, and he actually identified the underlying cause of this disease, which is the JAK-STAT pathway. He was able, during his study, to off-label treat 7 patients with JAK inhibitors and recovering them from certain death. This is basically a breakthrough which has never been seen before and gives hope for many patients out there in the world. Now it's my pleasure to hear from Thierry on his own what his motivation is on his research and what he thinks how the future looks like.
I'm here today to interview this, of course, about your research. Maybe you can tell us something about your latest research or LWT, yeah?
Sure. So maybe the core focus of our research, my passion, actually lies in bridging fundamental biological insights with patient care. And our lab here focuses on using cutting-edge technologies, mainly spatial proteomics, to analyze complex molecular disorders on a very fundamental, basic way. And we try to combine this with clinical expertise. The long-term goal is always to help the patient. And as a dermatologist, I'm particularly interested in skin disease, but I think the skin is a beautiful disorder as a model organ system to study many complex molecular alterations occurring on a tissue level that we can leverage to better understand biology and health and disease.
And why did you pick DVP, so Deep Visual Proteomics, as your method of choice for the analysis?
Coming from the clinic, I actually saw a couple of patients with toxic epidermal necrolysis. And it's really a devastating condition. So basically, it's a very acute blistering disease of the body that gets triggered by common medications in previously healthy adults or even children. And this really causes a detrimental effect. Up to 30% of the patients die. And even those that survive have long-term complications such as chronic pain of the eyes and mucous membranes, and of course psychological disorders that come with it. So I really thought we must change the way we address this disease, because at the moment there was no current standard of care. Every hospital treated the patients differently, and the outcome overall was quite poor. And so I reasoned that by joining the lab of Matthias Mann, being able to do molecular analysis with such patient-ish biopsies, we could actually help understand the disease and define new therapeutic targets.
Okay. And to do this, you picked basically DVP, so Deep Visual Proteomics. So why did you pick it, and what was the success factor behind it that you actually succeeded finding a cure finally?
Toxic epidermal necrolysis is a very rare disease. It only occurs in about 1 to 2 per million per year. So we needed some sort of technology that would allow us to get access to all those patient biopsy samples that are archived in all around the world. So we needed some sort of technology that could use FFPE tissue sections. Another reason was that we needed a technology that could, unlike traditional bulk methods that could only analyze bulk molecular alterations, we needed something to be able to dissect the tissue and look at the cellular players in more detail and resolve these molecular atlases on a cellular level to really understand what's going on in such a complex disease. Deep Visual Proteomics really combines those two. We can use your standard routine clinical FFPE tissue sections and still go after specific cells, for example, immune cells or keratinocytes, that play a major role in toxic epidermal necrolysis.
And cutting basically tissue layers is not enough because the immune cells are somehow inside. So how did you distinguish between the immune cells and keratinocytes?
Deep Visual Proteomics is a method where it's a very multimodal method. We use these tissue sections, we cut them out to special membrane slides, and we use imaging methods. It can be standard routine imaging methods such as hematoxin and eosin or immunofluorescence to actually define the cells of interest. So that's exactly what we did in these patient biopsies. We cut them onto these sections and we stained for immune cells and keratinocytes. So we were able to, even if they were very dispersed across the tissue in these biopsies, which actually really is the case in this disease, we were able to kind of collect them throughout the tissue using Deep Visual Proteomics. Now, after we stain for those cells, we know exactly where they are in the tissue. And we use that information, kind of like our GPS coordinates, and we take those slides, we go to the laser microdissection system, and we can precisely, in an automated fashion, cut out these exact cells from that tissue. And we use this for ultra-sensitive downstream proteomic analysis, which gives us basically an unprecedented insight into what's occurring on a molecular level in these cell types, even in these very, very long archive tissue biopsies.
Nice. And that way you were basically able to identify the root cause of the toxic epidermal process.
Exactly. So we looked at the different cells that we cut out keratinocytes and different immune cells. And what we saw in comparison to other inflammatory diseases also caused by medication is that immune cells and largely macrophages have a very high signal of the JAK-STAT signaling pathway. That is a pathway that is driven largely by interferon, but also by other cytokines. And luckily for us, there were already drugs approved on the market to target this exact pathway. So when we saw this data and we saw how overactivated this pathway was, and we knew how hard to treat this, that we cannot really treat this disease very well, we knew that this is a missing link, and we did everything we could to actually follow up on this. And in vitro and in vivo mouse models, we really proved that this was a driving force in this disease, and that by using JAK inhibitors, we can actually inhibit this reaction. And of course, knowing this data, we actually treated the first 7 patients worldwide consecutively with JAK inhibitors, and all had a very, very beneficial effect. So that was very satisfying, both on a scientific level, but also on a personal level.
I think so too here. All ended in a great publication in Nature, which also contributed that basically spatial proteomics was picked as Nature Method of the Year last year. So I guess we can be also very proud of this. And in connection to this, how do you see the future? Will DVP contribute to even more success stories like this?
So of course, I and we in the lab of Matthias are extremely proud to have contributed, I think, significantly to spatial proteomics being selected as Method of the Year 2024. But there are many flavors to spatial proteomics, and I think every single contribution has made this method the Method of the Year. And of course, most satisfying is that we actually contributed to bringing this method into actual clinical care. So I think that's where it really shines, In comparison to other spatial modalities, we analyze the proteins, which are really the molecular effectors of the cells. So I think if we target those, then we really have a high chance of kind of transforming the way we do science for medical purposes. And yes, I do believe that the range of diseases that we can understand better through Deep Visual Proteomics is basically unlimited. We'll have a totally new view of what's occurring on a protein level in an unbiased manner in the cells that the pathologist thinks is most important, or maybe the scientist has interest in. It's a whole new toolbox, and it's so modular that even further implementations such as multiplexed immunofluorescence will even further highlight the diversity of cells. And so it's a hot topic, and I'm happy to be part of it. And I think the future is really bright for Deep Visual Proteomics.
I think the same. I'm very happy that we hold. Was also a single piece in the past for contributing with my expertise in laser microdissection, and also and counting on you and your peers to improve the methodology and the overall workflow to make success like this.